Thank you very much. So microphone is on. Everyone can hear me. No noise. So actually, I'm not from the University of Lisbon, but that's okay. I'm from this completely okay, unpronounceable name, which is the name of a foundation in Portugal. So first of all, I want to thank the organizers for inviting me to be here. It's been a great meeting, actually. So it's, I think, right on the range of meetings where I don't understand enough, but I know enough that it's curious, and I'm not just into an introspection trip all the time. So it's the last day. People are maybe a bit tired. I've been adjusting the talk pretty much every day, depending on the talks of the day before. So what you're going to see is largely influenced by yesterday's talks, where people were showing some very preliminary things. So and then Orkun was saying that we need more non-equilibrium thermodynamics. So I'm going to spend a bit of time talking about that. And I want to say that most of the work that I'm going to present is work from Tommaso Cosetto, who was a postdoc in my group in collaboration with Jonathan Rodenfuss, who is a PI at MPI-CBG. And the very work in progress things is work that has been done by Camila, who is a PhD student that is somewhere around here in the audience, together with Shashi Tuchupalli at NCBI. Okay, yes, the topic is cellular growth as non-equilibrium thermodynamics. Okay, so as a physicist working in biology, my background is maybe a bit different to other people in the audience. So I have worked in the past on non-equilibrium thermodynamics of different subcellular processes, like signaling and error correction and proofreading. And at some point, I got very interested in how this type of formalism of non-equilibrium approach can be applied to metabolism and microbial growth. And when I talk about metabolism, I'm just going to be seeing it in this very zoomed out description, kind of like Avi was mentioning the other day, right? So it's an exchange of chemicals from the environment, which result in more biomass and some metabolic byproducts. So when I got very interested on this, I started reading different articles and different books. And I'm going to give you an extremely biased selection of the articles that I read and I found interesting. I mean, there are many others, but these are some of the quotes that I found quite curious. One of them has already been pointed here before, is from this review from Urs von Stocker, in which he describes how different microbes can dissipate as they grow, partitioning this dissipation in different ways, right? So some may produce more, exchange more chemical entropy, so may produce more heat. And he even referred to some evidence of endothermic growth, which I found quite fascinating. Somebody I don't know. Somebody was talking, Elizabeth, yeah, you were talking about this the other day, right? Then there was also this several papers from these are all very nice papers, no? The book from Royals, papers from Heinen, another from Claire Bezin, very nice sort of review article, in which there were different claims of how the dissipation in microbial growth could be related to biomass production. And there were different empirical formulas, so some like this, that if you're a physicist and you see a formula like this, you really want to know how this was produced or regressed or based on data. And then there were some quotes which were a bit cryptic to me. And then there was this claim of a universal value for dissipation per carbon mole, right? And the evidence for this, at least to me, it was hard to wrap my hand around it. And there was, of course, this fantastic book from Harold Morovitz, Foundations of Bioenergetics, which for a large part of the book, it's a normal thermodynamics book, I would say, of a good thermodynamics book. And then towards the end, talks about bioenergetics, thermodynamics applied in biology, and he goes to great extent into doing a calculation for the enthalpy and entropy of formation of biomass. Yes.
A quick question. I mean, this trend which you mentioned, which way does it go? So the more dissipative, the slower is the biomass yield.
So we're going to talk about that. So I think you talk about this.
Read it from this complicated model.
From this text.
Yeah.
So essentially here, the idea is that the dissipation of biomass can be related to the degree of reduction. And this is just a short quote from that paper, but we will show a bit of data on this. Okay, so this is sort of the background of the literature that I encountered. I'm going to give you is a very brief, superficial overview of the things we've done about it. And what I want to then emphasize a bit more is the work that is very preliminary. We don't have a paper. We don't have a preprint. We have nothing. So please don't scoop us, right? But I don't think you will. I don't think you will. And this is being recorded, by the way. So we all know each other now. But just to get feedback on it.
I already don't understand the hidden chemical entropy.
Yeah, so the next time we're going to clarify this, right? Yeah, so the first thing well, I went a bit quick over the questions, but the first thing we need is some kind of model, some kind of framework to describe this, right? And I'm going to say something like what ActSheet says, so as far as I'm concerned, this is not my framework, this is the framework. In the sense that we're kind of writing in physics terms, right? In a way that I could wrap my hand around what in the bioengineering community people know and have been using, right? So essentially, imagine a population, a synchronously dividing, growing population of cells. You can a steady exponential growth. You can describe this system through an influx of free energy, a production of biomass, and some outflux of metabolites. Okay. Now, under conditions of steady exponential growth, okay, under steady state conditions, if it's not steady state, what may happen actually is that the composition of biomass is changing, and these equations become more complicated. You can write them too, right? But they become more complicated. You have a statement of mass conservation. So this is similar to stoichiometric conservation in flux balance, right? But in this case, it's elements. So it's we're just describing the boundaries of the system, right? So what this means is that the flux of different elements that goes inside the system has to be balanced by the flux of those elements that goes outside the system, where CK is the composition, stoichiometric composition of biomass. Then you will have a statement of the first law of thermodynamics, which says that the heat flux equals the balance in enthalpies. So amount of enthalpy coming in minus the enthalpy going out. And there will be some enthalpy that is fixed into biomass. And then you have the second law. I actually forgot the second law. There's a greater or equal to 0 here. Entropy production is always positive. But essentially, this you can think of it as an entropy balance. So it says the entropy production rate has some term due to heat exchanges and some term due to chemical exchanges. Okay. So these chemical exchanges, you can see here, is when you partition the chemical potential of a chemical, it will be an enthalpy contribution and entropic contribution. So what you're putting here is a flux of this entropic contribution. And yeah, so this is quite general in the sense that we're not assuming any particular form for how the fluxes of the system depend on what in thermodynamics you would say the driving the forces, right? So we're not explicitly saying how these fluxes may depend on chemical potentials of the reservoirs, let's say, so on the concentrations of chemicals outside, or on internal variables of the system, right? We're not saying anything about that. So obviously, so this is almost not a model. I mean, of course, there's assumptions, right? You need to say it's growing, synchronously dividing, and so on, right? But yeah. So once you have this, you can write the quantities in terms of this framework, which I think, at least for me, I feel more familiar with. You can write the quantities that you find in these papers as this. This will be the biomass yield. This will be the dissipation per electron donor, the delta G, and this will be the dissipation per biomass grown. Okay, and these are the quantities that are that are often used as delta G of reaction and of biomass reaction. Okay, so once we had understood this, right, right, there was a choice, right? So we could focus more on looking at data, or we could focus more on the model. So what we choose is look at the data. No, there were very interesting statements. So we, yes.
Per ED, what's ED?
Electron donor.
Oh, electron donor, I see.
Yeah.
And where is the product inhibition here? You have the products, which of delta G's will be changed if product accumulates? I'm trying to map it onto what I know.
Oh, okay. So this would change. So here there's no accumulation, there's constant flux, right? So, but this is what I put here, this is the product. And these are the products.
Concentrations of products or fluxes?
This is fluxes multiplied by molecular enthalpy or molecular entropy of those chemicals. And the system is assumed to be chemostatic.
What's rho sub C?
Oh, sorry, yes. This is the carbon content of biomass.
And why do you separate this dissipation per ED versus dissipation per biomass?
These are the different quantities that people have used
Oh, different people, right?
in the literature, right?
They're not like, they're not separate because it's per ED.
Yeah, they're not separate, right? So in principle, these are the different quantities that, so I would say this is fundamental, and these are observables that people have defined in the literature.
Sure.
And we're gonna look at. Okay.
Is one more okay? Just to follow Sergei's question.
Yeah.
These molecular entropies, do they contain the concentration dependence?
Yeah.
So they're in the, okay, so
Yeah.
that's where the concentration.
Yes.
Just not explicit.
Oh, sorry. Yes. This will contain the concentration dependence, the log term of the chemical potential. Yes. Okay. So what we did is, let's see, we're doing with time. Okay. So what we did is we curated a data set, right? So we looked at what the evidence in these different papers was, right? So there was data in different of them. And we went to the sources of this data, and we annotated for each of these source papers the macrochemical equation, which is sometimes called referred to in the literature. We annotated the yields that had been reported, the yields that had not been reported. We completed with element conservation. Okay, so part of the data is incomplete. We annotated the pH, the temperature, the growth rate when reported. Some of the data was done in a calorimeter. Some of the experiments were done in a calorimeter. And then we also annotated, we cross-referenced, let's say, with chemical potential of the different involved chemicals, like CO2, oxygen, glucose, biomass, which is a big unknown. And that's what I would like to focus on. And then you can compute these quantities, right? Delta H, delta G, delta entropy exchange, biomass yield, which are the ones that are used normally to make these claims in the literature, right? So essentially, we looked at some of these papers that already contain a bit of data. We went to the core sources, we repeated the analysis, and then we increased the amount of data, essentially. And as one first validation of this data set, or not validation, but just to give you an idea of the uncertainties on the data, what you find in the x-axis here is what is the delta H, so the predicted enthalpy balance, right? So the difference in enthalpy between the fluxes in, fluxes out per mole of electron donor. And what you have here is the heat measurement for the experiments that were done in a calorimeter, okay? And in principle, these two things should match, should be identical. And they're not, but they're close. This is a log scale, so very important. You see sometimes there's quite significant deviations. And this is just to give you a notion of the type of data we have. So there are different types of bacteria. We have some archaea, some eukaryotes, respiration, fermentation, and a large variety of electron donors. By the way, this data set is public and annotated, so you're welcome to give it a look.
Is there chemoautotrophy in there as well, or no?
I think we do, yes.
I thought electron donor and acceptor had to be paired, right? Because why are you? Oh, I see. Now I see. It's simple versus carbon.
Versus. So there's a lot of different. Yeah. Okay. So then the first thing we checked is this statement from von Stockar, which was based on some evidence that there was endothermic growth, and different types of metabolism could be dominated by heat exchanges or by chemical entropy exchanges. And this is what you find, right? So all we did is we divide this delta H by T delta S and this delta SX by delta S. So in principle, you can essentially what you're plotting here is the contribution of enthalpy versus the contribution of entropy exchanges. And you can do just scatter plot, very simple. And all the data should be on this region, because otherwise they would violate the second law, right? So this was not imposed a priori, but the system is so simple that most of the data, we only think had maybe 3 entries, which we remove from further analysis, that would fall into this region. So you find there's data all over the place. Make of it what you wish, right? In particular, there's some. I guess what's interesting is you find respiratory metabolism is mainly heat dominated, right? Which is also what had been stated before. Fermentation has a significant chemical entropy contribution, And there is some data that falls into this endothermic region, which I think is quite cool. Of course, that doesn't mean this data, there's a lot of inaccuracies in this data set, right? So this doesn't mean that this data needs to be endothermic. For that, you will have to do those experiments and see that in fact that's the case. There is one paper, right, that was cited already before, in which that was reported, right? So if you look at the delta H in this paper, they coincide with the Q, or at least they coincide in the sign, right? So they're off, but they coincide in the sign. So this is the, let's say so essentially what we're doing is we're curating the data that people have been using over decades, right? And putting it together to analyze some of these statements. Then the next statement that there was is that there is a universal constant. I don't know if you remember, there's a universal constant of 500 kilojoule per carbon mole in the delta G of biomass. So I wouldn't say that's a universal constant, right? There's a log plot, right? But I highlighted here what happens for 500 joule. So how much of the data falls at 500 joules per carbon mole, factor of 2 up, factor of 2 down. So it is true that is, at least to us, was quite remarkable that you have a lot of data in this relatively small range of dissipation, right? So it suggests that maybe you don't need these complicated formulas, but as a rule of thumb, knowing that 500 kilojoules per carbon mole of biomass is how much this microbe dissipates is a good statement.
And what was the original sort of motivation for, like the original sort of basis for that statement, the 500 number?
I think with empirical as well, empirical.
So what is the x-axis?
So the x-axis, this should be, I have to say, I just did this plot. So this should be the different
nutrient sources,
electron donors, yeah. And here,
need both glucose and oxygen, but usually if you is it aerobic respiration? Is it?
I think that the actual legend is here. Right, so here we say so yes, all of this is aerobic respiration. All the circles are aerobic respiration. So you can see here, electron acceptor, oxygen. So all the circles are aerobic respiration.
So the x-axis contains acceptors as well.
Yes, this is what we call metabolic. So, okay, so this is essentially metabolic types, as we refer to in the paper, because there's a lot of, so we have about 50 or 60 different microchemical equations. So we group them per color and symbol. I'm happy to share this and discuss more.
Would anaerobic be significantly lower here if they were included?
So we have an aerobic.
Oh,
right, we have an aerobic.
And yet it's still this
within this range, right? I wouldn't say it's a universal value. And I should say this is again a meta-analysis. So
Okay, so and how big are these numbers compared to the overall delta G? Like if you total products and total substrates and total products.
It's
like
smaller.
The magnitude of this delta G.
Smaller. Okay, so I'm gonna try to, so I'm almost on time. I wanted to show you some of the work that was actually more preliminary and in
Maybe can you help us to put it in a framework for instance, Alfred outlined the NITP and so on. Just maybe it's a wrong question, but that's the only way how I'm kind of wired.
Right. So I know. So this this in some sense I think is agnostic to this. This is just a difference of what goes in and what goes out.
But the dissipation is exactly what is not converted. When you have two molecules, some of it converted to ATP or whatever useful forms, dissipation is what is not converted. So your y-axis is what has been dissipated per mole.
Of biomass produced.
Right.
Yes. So you take what goes in minus what goes out. You divide per what's produced. So it's different. It's a different. So it's more like the delta G of biomass. I think that's how they call it in the bioengineering community. So it's different.
We can call it a cost of producing 1 mole of biomass.
Yes, you can call it a cost. That's how we call it, and I think people call it. No, it's the cost of producing one mole of biomass. Now, I think what we want to do next is having some, seeing how to balance these numbers with some toy model of the type of that Avi did, and the people are doing of basic metabolism. And people have also done this in the past.
Maybe I'm the only one who doesn't understand, and
No,
then
no,
I
no.
can shut up. But I see that other people also have this question. So this is dissipation. So if I divide it by KT, I will get sort of Boltzmann factor. How biased is a given reaction, right?
No.
No? Then it's not the dissipation I know.
You mean how biased is this? This is microbial growth, right? So I think I'm not 100% sure that, so actually you were saying like the ratio of the flux in over the flux out equals exponential of the delta mu of the chemical reaction. So it should be that.
That's a dissipation.
But yes, but just that I don't think that's general for microbial growth. So that's for elementary chemical reactions. I think it's a true statement, right? I don't think that you can just say that the influx of chemicals is there over the outflux of chemicals is directly dictated by the difference in chemical potentials when there's a microbe in the middle.
Well, if there is one reaction inside a microbe, let's say
inside a microbe,
cow microbe, which has some
spherical microbe, I don't know, but let's say if these were just chemical reactions, right? Elementary chemical reactions, then what you're saying is completely true, right? So if this is just if microbes behave just as an enzyme, let's say, yes. But I'm not saying they don't, right? I'm just saying I'm not sure that you can make that statement so easily. I think that's a model, right? That's saying how the input fluxes depend on outside concentrations, right? That's a statement. Right? That's a statement. There's a detailed balance statement at the whole microbe level, right? I'm not sure I'm ready to put my,
Yeah, okay.
But I think, yeah. Okay, so I think essentially I'm already at 21 minutes. So I'm gonna maybe in very little time just mention the things that actually I wanted to spend a bit more time on, but that's okay. If that's okay, can I spend 1 minute since there's been some discussion? Yeah. So the last thing we wanted to discuss is the enthalpy of biomass, right? This is an important quantity, and it's a quantity we really don't know very well, right? We really don't know very well. At least in our data set, normally we need to put something here in enthalpy balance, but what number you put in there is just not very clear. So what we did is we did something of what we found in the literature, which is people have empirical regressions based on some data, based on the elemental composition of biomass. But the support for those was, we haven't, whatever. We took like 20 data points. It seemed to be some correlation. But when we put our paper, so that's the term. So Abby wrote us this. And I mean, he wrote a very long email, right? And we discussed. And we said the elemental and energy content of biomass is sparsely measured. Do you have any thoughts, comments on this issue? And we said that this is perhaps the most frustrating. We would like to investigate the energetic content of biomass. So I reread this email the other day. So what we did, what we've been doing, or Camila has been doing, is collecting whatever is known about this number from the literature. And the way you would, so these are her slides, change of color. So the way one way you could go about this is if you burn biomass, you will get some heat. You could write a combustion reaction, and then in that combustion reaction, you should be able to isolate the enthalpy of formation. So how does, in practice, people do this? They take a plant, for example. They would dehydrate it and annotate the water content. They would burn it, annotate how many ashes it produced, annotate the heat, and measure the elementary composition of the gases. Okay, so essentially what we were looking at was whether empirical relations that people have been used are supported by data of this type. Okay. And what that suggests is that you can somehow decompose biomass into, these are not functional sectors in any way. No, this is just a way of describing the data that we're collecting. There should be a water part, some combustible part, and some ashes left. And the combustible part should be composed of these 4 elements. So I just want to finish now because it's already 24 minutes, so that's too long. And I just want to show you the type of data that she's been collected. So I mean, in principle, we were interested in microbes, bacteria, which should be here. But she went a bit overboard and collected a lot of data of small animals and plants and fungi and other things. Well, I guess not many other things left. And at least we understood something. I should say these are small organisms, right? So in addition, she's collected data of tissues for large animals, which I'm not showing here, and of small molecules like proteins, amino acids. And I just want to point a small fact that we realize. I mean, it's kind of trivial afterwards, but well, there's some level of variability, right? And in the literature that we're using, often this notion of dry weight, but what you realize is that the variability gets reduced if you, so both these conventions we're using to report the data, so per dry weight or per ash-free dry weight. And so we realize that the variability reduces when you use ash-free dry weight, which is kind of trivial. It just says the part that actually burned, which is the ash-free part, is the one that produces the amount of heat, right? So anyways, so this is just something we realize. And I can just tell you at present what we're doing is we're trying to explore the remaining variability by looking at data in which was supported the lipid mass fraction or the carbon mass fraction. These are the kind of empirical relations that we're looking into that have been used in the past. Okay, so I'll end now, and these are the references and the people involved. And if you have more questions, I'm happy to take them.
Questions, maybe first from the Chinese.
Yeah, super interesting. Could you, so you had the slide about like delta G and aerobic respiration is sort of near this like
yes,
like limit that people predicted, you know, some range around it, but then some things are like an order of magnitude off.
Yes.
Can you just like wax poetic about what those things are and like why they would possibly deviate so far?
So here we have, these are hydrogenotrophic methanogens. This is a type of fermentation. I don't know. So I think one problem, right? You also have to take with there's also the question of the different data sources and variability from group to group and experiment to experiment. Okay, so before I can put my hand, so I can do what we're doing now to address some of these issues, right? What we're doing now is we're collaborating with an experimental group where we're trying to do all the measurements. They're trying to do all the measurements carefully, including enthalpy exchanges and entropy exchanges in yeast for respiration and fermentation, and be able to assess the variability there.
I mean, just briefly, could this relate to what Matteo was saying about like basically efficiency as a function of growth rate?
In the data that we collected, we define a thermodynamic efficiency and we plotted it as a function of the yield and the growth rate. We didn't see any clear correlation. That's all I can say.
Thanks. This is amazing because all week we kept saying, we keep using this delta G dissipation from the paper of Heinen. We hope somebody would measure it.
We haven't measured it.
Well, somebody would.
This is just a collection of data, including some of his data. This is a compilation of all the data that has been reported, essentially.
Right. Do you know if this value depends on the growth regime? Should it depend if it's actively growing cell or if it's starved?
I presume very strongly so, right? All of these are under condition of steady exponential growth. The other thing that we want to look into, well, you know, in the future, let's say, so we have in this experimental collaboration is how can we measure maintenance dissipation, right? Normally what people do, which many of you may know, is look at how this, for example, heat production rate depends on the growth rate and extrapolate to zero growth rate. I think that's based on this called the Peirce equation. Instead, what we want to do is, and our collaborators are beginning to do is in a calorimeter, have yeast in a situation in which is not growing, measure what is the heat flux, and then also measure the depletion of nutrients along this time to get an estimate for the entropic chemical contribution.
Would the dissipation be an order of magnitude different?
I cannot say. I cannot say.
Okay, so if I read this delta GV as the delta G of dissipation of the Heinen method,
yes,
right, that I can do, I think, then it affects the growth yield, right? So then you can read it, and of course the growth yield is variable. If you only think about it in terms of delta G of catabolism, delta G of anabolism plus delta G of dissipation.
Yes.
But it doesn't vary that much. So I can have a different growth. I don't have so clear that this number will vary with the growth rate. This is what I'm trying to make as a point. Because I have been working with this empirical formula of Heinen for a bit, and it seems to work quite well when you don't know too much about the parameters of growth of a microorganism. It seems to fit quite okay, you know? So it's a very coarse-grained approximation, but somehow it is empirically working. So I wonder if you think that Heinen formula could predict these numbers that you see here, or where it will fall, this graph that it creates with the number of carbons of the carbon source. And if you think that we need a new empirical formula that is as simple as connecting this delta GB with the number of carbons of the carbon source and the degree of reduction, or we will need any other parameters.
Since somebody else is starting, we can talk afterwards.
I
can go on. Yes. Do we need another empirical formula? I think so. People have provided these formulas, right, as a way to predict yield.
Yeah, which you don't know so much, they are useful.
What?
When you don't know so much.
They are useful, yeah.
Yeah.
As an order of magnitude.
Yes.
Yeah, I think we just, if you need an empirical formula, I guess the right way to do is do a regression on all the data that is here and try to see what's the best that one can get.
It would be interesting to see if Heinen's formula contradicts.
Yeah. Here we haven't used any of these empirical formulas, right? But yes, we can do it. Yeah.
Thanks, Bob.